ago. He came from the Albert Einstein School of Medicine, where he got his PhD, where he did the computational structural biology PhD. So this was a shift from him. He's been working with Mark and Peter on the CST collaboration, and he's developed a tool called KIA2 that he's not going to show today, but this is a new and improved version of the human kinome enrichment analysis and visualization. Today he's going to talk about Clustergrammer that started as a very small project, and Nick really took it off, and now we're using this Clustergrammer and component, visualization component in many of the tools as Gregory showed.
Hi everyone, thanks for having me. So I'll be discussing Clustergrammer, web-based visualization tool for making and sharing interactive cluster heat maps. So I might show a little bit on the browser, but I'll probably don't want to have it like go bad. But anyway, so basically Clustergrammer, it enables users to easily generate interactive and shareable cluster heat map visualizations, and a user can do this in several ways. One way is to go to the home page and upload a matrix of their own data. So here you can see. Anyway, yeah. So on the right side, you can see a screenshot of the Clustergrammer homepage where it shows a demo of an example heat map that you can interact with. You can also press play, and it'll walk you through some of the interactive features. And underneath it, it shows you the file format that you can use and where you can actually upload it and obtain a permanent and shareable interactive visualization of your matrix. So in terms of how the app itself works, it's a pretty simple app. You start by uploading your data, the matrix shown there. You can do it through the home page, you can use the API. And that data is then sent to the backend server, which is Apache server made using Flask. So basically what that does is take your matrix and cluster it and compute like various views of your data and saves that to MongoDB, and then you are going to be redirected to a web page that shows your visualization. And the front end is built with a JavaScript library called D3, which is very customizable. And I guess I'll try and see if I can maybe. Okay. Anyway, well, you can go there to see more of the interactivity, but I'll kind of show some static screenshots. So the matrix itself, what sort of makes Clustergrammer unique is that it's like highly interactive and has a lot of ability for you to explore like the high dimensional data within your matrix. So the core like idea is that there's like more than one meaningful way to look at your data. So I have here sort of a diagram of some of the interactivity. So you can zoom into your matrix, which is pretty important if you have a large amount of data where you can't actually read the labels. You can also reorder your matrix dynamically. So you can reorder it based on cluster order. You can reorder it based on the data within there. So you can sort the rows and columns based on the sum of the values in the row and column. You can also dynamically double-click the labels and reorder the matrix based on the data within a single label. And also some of the other features that are pretty unique that you won't find in other programs are like filtering. So you can like filter out rows of your data that have low sum value, for instance, to sort of filter out some of the noise, and to help you get to the data in your matrix that is particularly of interest to you. And there's some other features that I won't go over, but you can see them on the Clustergrammer homepage demo or in the help documents. Okay, so one of the motivations for working on this project is basically that visualizing the matrix is a pretty general problem that we run into quite a bit, and that other people are bound to run into. So we've ended up finding quite a few uses for it in the Mind Lab, and some of them are shown here. And I'll go in detail over two of the examples. Okay, so I'll just go over a couple of examples in a little bit of detail. So you heard about the Geneva app from Avi and Greg just a minute ago. And with this, the Geneva app is to just quickly summarize, it's giving you a report on gene signatures have like similar background or something similar about them. So in this case, I'm just showing you a visualization that's available on Geneva using Clustergrammer, and it's giving you a report on ALS gene signatures. So what you can see here are the differentially expressed genes in ALS, and so the genes are in rows, and the gene signatures are in columns. And so in Clustergrammer, for instance, it's initialized to show you the top 50 genes, but if you want to see more genes or fewer genes, like in this case, I switched over to filtering out the data to only see the top 10 differentially expressed genes. So it just allows you to quickly filter the data that's most interesting to you. Another example of some data that you can visualize with Geneva are the enrichment vectors that Greg was mentioning a little earlier. And this just kind of highlights some of the customization that you can get, in the sense that what this is showing you are the rows are enriched terms for the gene signatures on their columns. And because the terms are enriched based on an up and down gene list, you could in principle have a single cell in your matrix be enriched for like up and down. So in the sense that you would need, you basically have two values in one matrix. So I'm depicting that visually by like splitting the tile. And that's sort of an example of a type of customization that you can get with Clustergrammer. Clustergrammer is also being used to generate dynamic visualizations for a tool that Avi mentioned before, and that Chana will be discussing later, called L1000 CDS squared. So this just shows you like a rather large matrix that you can visualize. So what this is showing you is the results from the search tool, L1000 CDS squared. So the way this tool works is that you input a gene expression signature, and it searches the L1000 perturbation database and finds signatures that either mimic or reverse your gene signature. And this allows you to visualize the search results and your input signature. So what this is a little bit complicated. On the rows, if you see the red and blue, that's showing you your input signature, so your up and down regulated genes. In this case, the search was for perturbations that reverse the expression of your signature. So what you'll see is that the cells in the matrix are blue on the top and red on the bottom. So they're decreasing and increasing respectively for up and down expressed genes. So you can zoom into that to see exactly what's happening. So you may have some information that would lead you to want to perhaps test a perturbation based on the specific genes that are involved. And this is just showing you how you can reorder the matrix. And like for instance, you may not want to choose the top ranked perturbations. You might maybe want to choose perturbations that represent like different clusters in your data, perhaps like have the best chance of finding a drug that's going to work for a drug combination. Yeah, and so Clustergrammer is available for developers who are interested in producing a visualization on their own. So there's a couple ways you can do this, but one way is you can use the RESTful API to obtain a visualization directly from Clustergrammer, so you don't have to deal with any of the code. But if you're interested in looking at the code itself, it's an open source project, which you can do on GitHub and that way. And also, yeah, just like to acknowledge the Maya lab, especially Greg, who helped out a lot with the development of the code, and a lot of the. Any questions?